Thanks, Dr. Finlayson. First of all, it's my great pleasure to introduce Bishwa Sapkota, who is going to be our next speaker. Bishwa joined my program as a PhD student in the fall of 2018 after his master's in remote sensing at the University of Arkansas, Monticello. His program is supported by a Dean's Strategic Assistantship. Over the past couple of years, Bishwa did a fabulous job applying the remote sensing tools and computer vision technologies for weed detection, mapping, and management applications. I would say that he truly made significant contributions to the science of weed science. He won several awards in student speaking and poster contests. He was selected as the outstanding graduate student by our department in the agronomy section, as well as he was nominated for the Gerald Mott Award by the CSSA. Without any further ado, I'd like to turn this over to Bishwa. Bishwa, take it away.
All right, can you hear me?
I can hear you, and we can see your slides.
Thank you, Dr. Muthu, for that sweet introduction. Hello, everyone. My name is Bishwa Sapkota, and today I'm going to talk about my PhD research, which is entitled Remote Sensing and Computer Vision for Weed Detection and Mapping in Agricultural Systems. So, as we all know that weeds are bad and weed can cause significant impact to the crop yield. In the US, the chemical application has been the most dominant means of weed control. And because of this dominancy, a lot of times farmers end up spraying herbicide injudiciously in the field. And because of this injudicious application of chemicals, we are seeing some kind of very dire problems like herbicide resistance issue in plants. And sometimes because of unwise use of this kind of herbicide in the field, farmers are having more expenses compared to the revenue, which is not what they want. So because of this, it's important for farmers to actually change their spraying strategy or resort down to different weed control methods. One of the methods that the farmers can use is site-specific weed management approach. So it is an approach in which the herbicide are sprayed or weed control methods are applied when and where it is needed. There are various technologies that have been developed today that could help the site-specific weed management. These particular technologies can be grouped down into two different approaches, real-time and post-mapping. In real-time approaches, a lot of the robot machines or tractor enabled with sensors are sent to the field and they are programmed to detect the weeds. And after they detect the weeds, some machines are designed to actually spray the herbicide over the detected weeds, whereas some machines are designed to actually use the mechanical cultivator and then pull and zap the weeds. So we have a different approach, which is post-mapping. In this approach, the weeds in the field are first mapped using the remote sensing platform like UAVs. And after preparing the weed maps, they are converted to prescription maps, which are uploaded to the GPS-enabled tractors. And these GPS-enabled tractors are sent to those areas where the weeds needs to be controlled. In this way, just using their resources very wisely and appropriately. So the major backbone of the two different technology that I just talked is the computer vision technique. So computer vision technique is a technique by which the computer reads the images and allows us to actually detect and classify the objects that are seen in the image. So since last 4 decades, there has been a huge improvement in computer vision approaches for weed detection. And this particular computer vision techniques for weed detection can be categorized into three broad techniques, I would say. The first technique is pixel-based detection, where actually an image which is composed of pixels are actually assigned into either falling into crop or weed and using some machine learning algorithms. And there is another, excuse me, there's another technique called crop row-based detection, where the crop rows are detected using the computer vision machines, and any vegetation lying outside of the crop rows are assigned as weeds. And that's obvious because anything growing outside of the crop rows are called weeds. So, and the third approach, which is very popular today is deep learning based detection. In this technique, the thousands of images belonging to weed or crop are actually fed into the neural network system, which then actually trains over all those images and then actually produce the detection model. So it is important to note that a single computer vision technique may not solve all kind of weed detection problems and farmer objectives. So every weed detection problem has to be tackled with unique computer vision approach. Because of this issue, in my PhD research, we thought like, okay, let's customize and evaluate different computer vision techniques for various weed detection and mapping problems. So in my PhD thesis dissertation, I had 4 experiments, but because of time limit, I'm going to talk about just 3 of my experiments. In the first experiment, so we had a wheat problem of early season ryegrass intermixed with wheat, and that was addressed with pixel-based detection. And for experiment 2, we had mid-season weeds in cotton, and we applied crop row-based detection method to solve the problem. And in experiment 3, which was where we had the early season weeds in cotton and soybean, we used deep learning-based detection approach to actually address that issue. Moving on to my experiment 1, which is detection of Italian ryegrass in wheat using pixel-based detection approach. As you all know that Italian ryegrass is the most dominant weed in wheat, and some studies have found that this particular weed can reduce yield up to 90%. Because of this reason, it's very important that we manage this particular weed in a timely fashion. So in this experiment, we actually had 2 different objectives. The first objective was to actually detect and map ryegrass. And the second objective was to actually predict the competitive effect of ryegrass on the wheat grain yield reduction using the mapping information that we could get from the computer vision approach. So this experiment was conducted in 2 different sites. In each site, we had training area and validation area. And in training area, we had 4 different ryegrass density treatment plots, as you can see on the screen. And this setup was designed in complete randomized block design, and all the density treatment plots were replicated 4 times. So for this experiment, we collected RGB imagery using Phantom4 Pro. We flew at 10 meters, and the resolution of the imagery that we obtained was 0.3 centimeter per pixel. And at the end of the season, we also collected ryegrass biomass, seed yield, and wheat biomass and wheat grain yield to see the competitive effects of ryegrass on the wheat. So moving to the method section for the objective 1, which is to detect and map ryegrass. The first step was actually to develop several features from the imagery, like vegetation indices and some color space transformations. And after generating these features, our next job was to actually select the pixels belonging to ryegrass and non-ryegrass from the imagery. And when we select these pixels, we can tell the machine learning algorithm that, okay, these pale looking green pixels are ryegrass and these bluish looking pixels are non-ryegrass. And after that, when the machine learning algorithm is trained, we can assign an unknown pixel to the machine learning algorithm, and that machine learning algorithm will tell whether that pixel belongs to ryegrass or non-ryegrass. So after we train the model and develop the ryegrass canopy coverage map, we calculated the ryegrass canopy coverage area from the map, and we use that area into the regression model where we could predict the effect of ryegrass on wheat grain yield reduction percentage. So we use simple polynomial regression for this approach. Moving to the results from objective 1, which was to detect and map ryegrass, we can see in the screen the true color imagery on the left-hand side and the corresponding detection map on the right-hand side. The red color areas represents ryegrass, whereas the yellow color areas represent non-ryegrass vegetations. So for mapping the ryegrass, we obtain an accuracy of 93%, which is pretty good. So moving to the results from objective 2, which was to actually use the image-derived information to actually predict the effect of ryegrass on wheat grain yield reduction percent. On the left-hand side, we can see a regression graph where the x-axis is ryegrass canopy coverage area in percentage, and the y-axis we have wheat grain yield reduction percentage. We can see that ryegrass canopy coverage area did a pretty good job in predicting wheat grain yield reduction with R-square value of 0.67. On the right-hand side, we can see the ryegrass canopy coverage map that we derived from our objective one. And on the downside, we can see a grid map that shows the competitive effect of ryegrass on the wheat in 1 by 1 meter grid level. So we implemented this, what we did was we implemented this particular model into the ryegrass canopy coverage map, and then we achieved this particular grid map. So this grid map will provide the information about the effects. So for example, this particular area would represent the area where the effect of ryegrass on the wheat reduction, wheat grain yield reduction is higher, whereas on the green area it is lower. So in conclusion, the ryegrass can be detected accurately using pixel-based technique. And we also saw that ryegrass canopy coverage area can be a good predictor of competitive interaction effects. So actually, we already published this paper last fall. The citation for this paper is actually shown on the screen. And if you would like to get a link of this paper, I'll be more than happy to provide you after this seminar. Moving on to my experiment 2, which is mapping and estimating weeds in cotton using crop row detection approach. In this approach, we tried to actually identify, detect, and map the mid-season weeds like morning glories, red spangled top, and palma amaranth in cotton field. For that, we set up 3 different density plots: low, moderate, and high density plots. These plots were set up in complete randomized block design. And after we set up the experimental plots, our next job was to actually capture the aerial imagery using phantom pour. We flew at 15 meters, and our resolution of the imagery that we got from the drone was 0.8 centimeter per pixel. So moving on to the image analysis, because this is the main backbone of this particular experiment. So our concept was to actually identify the crop rows in the imagery. So what we did was we implemented series of computer vision techniques like canny-edge filtering to actually pick the prominent points of the crop rows, as you can see here. So those prominent points can act as a fuzzy line. And then we tried to implement a half transformation method to actually connect these fuzzy lines to a solid crop rows, as you can see on this third figure. And once we detect the crop rows, it was easier for us to actually assign every vegetation lying outside of the crop row as intra-row weed detection, intra-row weeds. And we also saw some instances of intra-row weed within the row. So for that, we used machine learning algorithm and detected some intra-row weed within the rows, as you can see in the blue color on the 4th figure. And finally, we merged these intra-row weed detection results with intra-row weed detection results to create a final classification map. Here's the results for the objective 1, which was to map the weeds in the image. So we can see the weed map that we obtained for all the experimental plots in our experiment. The weeds are shown in white color, whereas the rest of the vegetation and background are shown in black color. On the right-hand side, we can see a graph that shows the mapping accuracy of this particular approach for different density plot in our experiment. We can see that the accuracy was higher for low density plot, whereas it was lowest for high density plot. So this graph generally gives an idea that, okay, the accuracy goes down when the weed density increases in the crop rows. So we had 2 conclusions from this study. So accurate weed coverage maps can be created with crop row detection approach in cotton. And we also saw that accuracy can decrease with increase in weed density. And we also published this paper already last fall. The citation for this paper is given in the screen. And again, if you would like to have this link for this paper, I'll be more than happy to provide after the seminar. So moving on to my last experiment, which is early season weed detection in cotton and soybean using deep learning approaches. I'm going to talk a little bit more for this experiment because this deep learning approaches is a very hot topic today and everybody is talking about it. So in this experiment, we are addressing a weed problem where we have weeds at very early growth stage in cotton and soybean that escaped the pre-emergence herbicide application. Now, our first objective is to actually detect these particular weeds and localize them. In other words, finding the bounding coordinates of this particular weed. That way, this coordinate information can be fed into the robots to actually target that particular weed. In addition to that, we also try to move further forward and then try to actually predict the boundary of the weed for canopy segmentation. And in our objective 2, we wanted to predict the biomass using the canopy area that we could get from this canopy segmentation. So to accomplish this, we used a very popular deep learning model called Max RCNN model. It is a convolutional neural network CNN-based model. It was actually developed by Facebook AI research group for their own purposes. Since then, this particular model has been used in several applications like MRI imaging, evaluating the surgery robots. And recently, this model has been used for few agricultural applications like latest plant counting and automatic cattle counting. So looking at these applications, we were inspired, and we also thought, okay, let's use this model for our research in weed science and try to detect the weeds. Just wanted to give you a very brief overview of how CNN works. So actually, a CNN model has hundreds of layers within it, and within each layer, a CNN model can produce different feature representation of a particular object. For example, on the cotton image here, the CNN model produces this kind of feature representation in layer 1, whereas it emphasizes a different feature representation in layer N. In this way, a CNN model actually produces thousands of feature representations and tries to learn all those feature representations to actually detect the weeds in an unknown image. So for this experiment, we collected aerial images using a Fujifilm 100 megapixel camera that was attached to the Helio drone, the drone that was produced in USA. And then so when we captured the imageries, we had the weed species that were present in the field were a tall morning glory, ivy leaf morning glories, and some grasses like Texas millet and Johnson grass. We also had some instances of Palmer amaranth and sickle pod, but they were very nominal in number. After we collected the imageries, we collected the biomass of morning glories and grass, and we collected 75 plants for each of these species. Moving to the methods for my first objective, which was to localize and segment the canopy, we actually implemented 2 different approaches for this objective. Well, the first approach was to use real images. So in deep learning, the main task, I would say, is to actually annotate the weeds in the image and label them either as falling into weed or crop. This is a very tiresome job. So in real images, what we did was we had altogether 850 images, and we had to go over each imagery and then try to annotate the weed and label them either falling into cotton, soybean, morning glory, grass, or other weeds. And on the right, you can see the sample result for the annotation. It took us around 250 hours to actually just go over each image and then annotate. This was not efficient. We are not happy with this. So we had to look for an alternative approach. So we borrowed a concept of a synthetic image. So what we thought was we were thinking of developing the real looking synthetic image using some representative cotton plants that could be seen in the real images. So what we did was we actually clipped some representative plants for all these categories from the real images and then pasted over this bare background by adding several random variations, like some of the plants were rotated, some were scaled, and some were changed with the color. And then finally, we produced as many number of images that we wanted as synthetic images. The main advantage of this approach is actually that we don't have to manually annotate the weeds. So when we generate the synthetic images, the annotation is done automatically. So we completed all this process in 4 hours, as opposed to it took 250 hours for real image based. So after we developed after we annotated the real images and we produced the synthetic images, our next job was to actually feed this both images and annotation to the Max RCNN model, and to develop the Max RCNN model for each of the approach. And then we implemented each of the approach over the test images to predict the weights. Moving to the results for my first objective. So on the screen, you can see two different graphs that shows the agreement between predicted number of weights and observed number of weights by these two different models, real image-based model and synthetic image-based model. On x-axis, we have observed number of weights, and on y-axis, we have predicted number of weights by these models. We actually tested these models over 50 test images, and this is the results that we got from there. We can see that real image-based provided better R-square value compared to the synthetic image-based, but it should be noted that synthetic image-based was really quick and it was time efficient, and still it still provided comparable accuracy to real image-based model. This is the results from both the models for soybean and cotton field. On the upper row, we have the results for soybean, and on the bottom row, we have the results for cotton. We can see that how accurately and precisely the real image-based model detected the grasses and morning glories. It not only detected, but also segmented the canopy boundaries so well. But if you look at the synthetic image-based model, we can see that some of the grass species are being missed by the synthetic image model. But still, we can see that synthetic image model still did a good job as compared to the real image-based model. And the story was similar for cotton as well. Moving into the results regarding the biomass prediction using the canopy area. So on the screen, you can see two different graphs for two different species, morning glory and grasses. On x-axis, we have canopy area in centimeters squared. And on y-axis, we have above ground dry biomass in gram. We can see that the canopy area that we derived from the canopy segmentation results from objective 1 was pretty good at predicting above ground dry biomass for morning glories and grasses. Among these two different species, we can see that grasses were better predicted using the canopy cover area from the segmentation results. So we had two different conclusions from this experiment. The first conclusion is that it is possible to detect, localize, and segment the canopy at individual plant level accurately. And we also saw that synthetic images can be a greater alternative to real images for detecting weeds using deep learning techniques. And we also saw that accurate biomass prediction can be made using deep learning technique. And among the broadleaf and grasses, the grasses may be better predicted with the canopy cover area that we developed from the model. So to summarize the findings of my PhD research, I could summarize my overall findings in 3 points. First is remote sensing can assist with site-specific weed management and help farmers to actually spray the herbicide very wisely. And one computer vision technology cannot fit all types of weed detection problems. So a lot of computer vision technology had to be explored to improve these site-specific weed management approaches. And among all the techniques that are available today, deep learning technique has a great promise, especially with the synthetic image concept. Now, there is a huge room for improvement for these site-specific weed management approaches. The improvement can be made over the computer vision technologies, and any future research targeting to help site-specific weed management should focus on computer vision technologies and try to customize various technologies that we have available today to solve new weed detection challenges. So with that, I would like to thank Dr. Muthu Kumar Bhagavathyanan, my major advisor, for providing me this opportunity. And also would like to thank Dr. Rajan Dr. Nolte and Dr. Popovsky for guiding me through my committee as being my committee members. Also would like to thank Dean's Strategic Assistancy, Texas A&M AgriLife, Texas State Cotton Support Committee, and Cotton Incorporated for providing the funding to carry out this research. Also would like to thank the web resources like Stack Overflow, Stack Actions, Towards Data Science, and GitHub for providing free of these massive web resources that I could utilize to use to develop my models for my PhD research. And last but not least, I would like to thank these lovely people that we can see in the screen for providing me help and showing cooperation over these years. Thank you so much, everyone. So with that, I would like to thanks, and then if there are any questions, I would be more than happy to answer them.
Great job, Bishwa. Very interesting. Very interesting. Questions?
I do have a question if you have a moment.
Yep.
Fire.
Yeah. So with these visual techniques, how adaptable would you say they are? So if you have a different crop growing, or if you have weeds that you didn't predict, how adaptable do you think these are to those?
That's a very great question. The question is actually how this computer vision model adapt to different cropping scenarios. So actually, these all techniques that I described today are supervised model, which means that you're gonna have to provide some training samples before you could expect the model to predict those crops. So if you are using your model, if you are training your model to detect soybean, you can expect that model to detect the cotton. So in that case, you would be needing to provide more training samples to the cotton. So, but with this deep learning technology that we have today, it's possible that we can merge many samples together and then can be used for broad applications like detecting weeds for various crops and various weeds.
Thank you.
Thanks.
I have a question toward the experiment 3. So have you ever tried traditional machine learning to classify, like pixel-based classification toward the images? Since from your example image, they from my experience, they are pretty good accuracy just using pixel-based. So deep learning, yeah, they probably will increase the accuracy, but that will increase like the cost and computer power. So if pixel-based classification is good enough for detect the grasses and the broadleaves, so I just want to know your reasoning to go to deep learning at the first place.
That's a really good question. Actually, these are the problems that we encountered when I started this project. So actually, this pixel-based detection, their purpose and the scope is different compared to the deep learning. So in our experiment, we used pixel-based machine learning or conventional machine learning to actually create the canopy coverage maps. We didn't expect the pixel-based. So actually, we didn't want the individual plants for the ryegrass experiment where we used pixel-based approach. So in that, we just wanted the canopy coverage. But if you want a single plant to be detected, I think pixel-based and conventional machine learning algorithm may not provide you a great result. And the ultimate solution that we have today is to go for deep learning. If you are trying to detect an individual plant, I'm not talking about mapping the canopy coverage or whatever it is. So this is the reason why I tried to use deep learning for my last chapter, which was to actually detect and identify a single particular plant. Does that answer your question?
Yes.
Yes.
Thank you.
I have a question for you. So my question is, what amount of training / certifications and licensing have you personally gone through to get to the point of not being able to fly a drone to now to the point of where you're taking imagery and applying herbicides and things like that? Just a very brief walkthrough of licensing / certifications on that.
So Mason, I think this is a really good opportunity for the beginners or whoever is interested in this technology to answer this question. So first of all, in order to take the images, we need to be certified by FAA. So we need to have a remote pilot license, right? I think for my experiment, that is the only license that I got. I'm not using UAV to actually spray the herbicide. So I don't have a herbicide license for UAV spraying. So other than that, I guess I have some remote sensing certificate, which allowed me to actually use this kind of computer vision technologies, which allowed me to have different computer vision knowledge. And so yeah, so it'll be just remote pilot license actually that I obtained to carry out this experiment.
All right, thank you.
Thanks.
I have a question regarding your altitude that you flew at. I saw 15 meters. Can you, I guess, explain why you chose 15 meters for your altitude? And I guess in experiences I've had, the lower you fly, the lot longer the flight takes. So is this something you're looking for a, I guess, potential grower usage down the road where you're flying a field, and if you're flying low, how long is that taking? Kind of what speeds are you flying at?
Actually, the reason for flying lower is actually to take the high resolution images, because see, we are trying to deal with detecting the weeds that are hidden under the canopy of cotton, right? So we need to have a really good high resolution imagery. So we could produce those high resolution images only by flying lower, because we are limited with the camera sensor there. Our camera sensor is not that good. So, and yeah, by flying lower, our flight plan may be longer, or we may have to fly a little longer, but we cannot trade off, our flight time with the amount of accuracy that we want. So we went lower in altitude and flew the drone. And for commercial purposes, it depends on the objectives. If they are not dealing with such complex problem, then they are good off by flying it higher and then taking low resolution images.
Okay, thanks.
Other questions?
Yeah, I have one. I was wondering if you evaluated or what sort of evaluation metrics you use for NASCAR CNN, and what would you get?
Yeah, thanks Matthew for asking this question. Actually, I was expecting this because of time limit, I could not report some mean average precision values that I calculated. Actually, these are the common accuracy metrics that we need to report for deep learning, but because of the time limit, I didn't report in this seminar. But yeah, for my deep learning approach, I use mean average precision from COCO style that are available in web resources.
Cool, thanks.
Other questions?
On that same project for your synthetic images, is there a way to improve your synthetic images just by doing more options? I mean, you had listed 4 different ways you rotated it, color change, et cetera. Can you just increase the number of choices there to improve your overall accuracy for that?
What a fantastic question. Thank you, ma'am. Yeah, that's definitely doable. We can add more variations, and we can diverse our training samples. Yes, that's definitely doable. But yeah, we just tried 3 different approaches for now. But again, with the synthetic image approach, you are not bounded with number of samples, so you can produce as many samples as you can. So our next step we are looking into is we are trying to produce even more number of synthetic images and feed into model to improve the accuracy. Yes.
Right. Great. Thank you.
Thanks.
Bishwa, there are also a couple of questions on the chat box. I don't know if you can look at it. The first one from Jeff. How did you assess the accuracy for ryegrass classification?
Okay. For ryegrass classification, the accuracy was measured or assessed with the help of pixels. So we selected some pixels of ryegrass and non-ryegrass and tried to, fit into the model and then predict the outcome of those validation pixels. And if our predicted level for those pixels were matched with what we saw in the imagery, then we would call it as true positive. So yeah, it was actually assessed based on the validation pixels of ryegrass and non-ryegrass. I don't know if that answers to the question.
I guess my main curiosity is who selected your pixels. Was it random?
Yeah, it was random. But one thing we have also mentioned in the paper is that, there is no limit, there is no standard how many samples do you need to select for validations. So we just went with 250 samples for validations. Some reviewers reported it as more than sufficient. And some reviewers said, it could be better by adding more validation sample. But because of the limitation of the ryegrass pixels in the area, and because of the time limit as well, we just went with 250 samples that were selected randomly.
Dr. Ibrahim, you got a question?
I have it in the chat actually.
Yeah, I could see it. I tried to reply, but I didn't manage.
Oh, no problem. Yeah, can you hear me?
Yes.
So, Sapkota, this is a very good talk. Thank you so much, and I like the visuals. So the best time to control Italian ryegrass is actually pre-emergence. And the window for applying it before the weed emergence is also very, very small. So, does this create a dilemma for the detect and apply, detect and control approach? Especially with regard to the herbicide option. And we're seeing a lot of ryegrass resistant weeds actually in the wheat. So that's another dilemma. What do you think about this? That's one question. The other question is the R square for validation of the model. Do you have that R square?
Yeah. Yep.
So is this the R square between the visual or the machine data? Is that the level 2 data or the visual ground truthing data?
No, this was the R square developed with the image derived area, image derived canopy area.
Okay, I understand that. What I meant is if you want to validate with ground truthing data, so what's R square looking like?
Oh, okay. So we have actually we had like five validation plots to actually to validate this particular model, but I didn't show in this slide because of time limit. But if I remember, it's around 0.6. The R square for validation is also around 0.6. So it's pretty much worked for that particular.
That is between the ground truthing data and level 2 data.
Yeah, between the ground truthing data.
Okay, and so that answers my question. But the first question is, as I said, ryegrass, the best time to control it is pre-emergence.
Mm-hmm.
And by the time you see it and distinguish it from the weed, it might be too late. I mean, I like the approach very much, but I'm telling you that there is a dilemma that for the detect and control approach, how do you address this issue?
Actually, that's a really great question, actually, because the ryegrass controlling is done by pre-emergence rather than the post-emergence.
Yeah.
But actually, our research was oriented in actually solving the problem if ryegrass emerge in the field. Now, the farmers apply their control measures, but sometimes their control measure doesn't work. A lot of time it doesn't work. So we still can see some ryegrass popping out in the field. And now our methods or our experiment comes into play. That's when we are trying to, now we're helping farmers to actually detect those ryegrass and then helping them to actually spray only where it's needed. Because if they go in a conventional way, they're gonna apply the herbicide again throughout the whole field. And we may not see as much as ryegrass that we could see throughout the field. And that'll be a whole wastage of herbicide resources. So to save that, we are actually detecting the ryegrass.
Anyway, it's a very good talk. Thank you so much.
Thank you, Dr. Ibrahim. Really appreciate it.
Other questions, comments? All right. Excellent talks. Thank you very much.